today I want to talk about a few different projects that I've been working on. Most likely I've got too much information in here, but now I realise that it becomes, as you do more research, it becomes very difficult to remove things because you'd like it all and it's hard to choose what to get rid of. So I had removed this in the interest of time, but I'm adding it on now. As Peter mentioned, Maren sort of introduced a lot of females through the lung group. This is the early lung group, and Sarah and I were the first lung chicks, I believe. So I was Maren's first PhD student, one of my main claims to fame. My other one is probably that I had the most children in the shortest space of time around here. So I had 3 children within 20 months. Has anyone else had more than that? No? So I might get the prize for that. So it's been very intense, and it still is, and one of the reasons I love coming into work. So when thinking about the 3 projects I wanted to talk about, I wanted to think about how they sort of relate to each other, and it's quite obvious it all comes back to smoking. So most of my research relates to smoking-related diseases and e-cigarettes, which is I'll talk about towards the end. So the consensus is out, smoking is bad for you, but just to emphasise that, I wanted to go through a few facts. So 7,000, that's the number of chemicals that are present when you inhale cigarette smoke, more than 7,000. So that's a lot of chemicals to be in contact with any part of your body, but especially in your lungs, which are a very delicate system. 1 in 10, so that's the number of deaths worldwide that are attributed to cigarette smoking, so 1 in every 10 deaths worldwide. 1 billion is the number of people that are projected to die from smoking-related diseases in this century. And nearly every body part is affected from smoking. So not just the lungs, that's what I'll be focusing on today, but every part of your body is affected in some way by cigarette smoking. And out of every 3 smokers, two out of them will die prematurely from smoking-related diseases. So these facts are pretty astounding. But what I find more amazing is the fact that a lot of people are still smoking, so everybody really knows about the effects. I mean, there's a lot of things that are bad for you, so I get it in that sense. But this map, it is 5 years old now, so things might have changed a little bit. But in looking at this, so the brown, this is the legend here showing the proportion of males over 15 years old that are smokers. So these dark brown ones are that 40% of males in those countries are smoking. And you can see there's quite a few countries with that statistic. In New Zealand, about 12.5% of adults smoke, but it's much higher in Māori and Pacific Island communities, over 30% for Māoris. In China, I added on here, so 28% in total, but 52% of men smoke. And I think the highest country had 78% of males were smokers. So just briefly to introduce how the lungs work, probably a lot of you are familiar with this. So your lungs are within your chest cavity. They have mechanical properties, so when you inhale, your diaphragm and intercostal muscles contract, which expands the volume of your lungs and brings sort of airflow into your lungs. We have these 3 different tree-like structures. That's a cast of the human lungs, the white showing the airways, the red the arteries, and the blue the veins. So these are tubes where the air and blood flow down to the gas exchange surface. This bottom picture here is a scanning electron microscope image of the gas exchange surface. So these are called alveoli, which is where the air and the blood come together. There's a huge surface area here, something like 80 meters squared. And the diffusion membrane is very thin, so you get exchange of oxygen and carbon dioxide here. So there's lots of things that can go wrong in this system when we come into different lung diseases. So some of the effects from smoking are indicated here, and I'm not going to go into detail of all of them, but the main diseases that my research has related to is to COPD, that stands for chronic obstructive pulmonary disease. And about 80% of these patients have a history of smoking, so there are other factors that may cause you to get this disease. It's the 3rd leading cause of death worldwide. So this is actually a CT scan from a patient with COPD. So one of the features of this disease is called emphysema, where you get a breakdown of the gas exchange tissue. So as well as minimizing the surface area for gas exchange, it also changes the mechanical properties of your lungs. It makes it quite difficult to get fresh oxygen into your lungs. Lung cancer is the other one, and probably the one that everyone is familiar with. So 90% of people with lung cancer have a history of smoking. And it's New Zealand's deadliest cancer, mostly because it's detected relatively late on, and because it's difficult to treat. So current clinical tools are fairly inadequate. So the main diagnostic tool used is called spirometry. It's where you have to blow into a device, and this measures the the flow rate or the volume of air that you can breathe out of your lungs. So the main measure is known, or one of the main measures is the forced expiratory volume in 1 second. So this is how much volume of air you can breathe out in 1 second, and that measure will come up a few times in the talk. So these measures, while they can detect if somebody has a respiratory condition, they're very non-specific. So it's very difficult to tell what's happening in their lungs, where the disease might be, and that sort of thing. So in our approach, we use computational models to bring together clinical measurements, imaging, both structural and functional, and experimental measurements. So we use the models to really bring these things together and increase our understanding of how the lungs are working, and ultimately to improve measures of lung function and things like predicting changes in lung function over time or after different interventions. So today, as I said at the start, I'll be talking about 3 different areas. The first one is lung cancer. So this is a project I'm working on in collaboration with University College of London. And specifically, we're looking at radiation therapy treatment for lung cancer patients. So this is just one method of treatment that applies high doses of radiation to remove the tumour. So there's a big trade-off with this sort of treatment. So while they irradiate and kill the tumour cells, they have to irradiate a much larger volume to ensure that they remove all of the tumour. So there's often a lot of healthy tissue that's damaged as well. So there's a trade-off between removing the tumour, but also removing healthy tissue, which often leads to reduced lung function. So in this way, we're trying to work towards improving the planning process of this radiation therapy treatment. So the main side effect of this is called radiation-induced lung damage, and it's a treatment-limiting toxicity. So this means that some patients either won't be treated, or they'll be treated with lower subtherapeutic doses. So they'll be treated palliatively rather than curatively because of the estimated risk of their outcomes. So the clinician basically uses a best guess to estimate the patient outcomes after this treatment. So they consider things like the patient's age, comorbidities, their baseline lung function, but it's often a best guess by the clinician. So there's no very accurate ways to predict which patients should be treated and the dose that they should be given. So some of the issues with this is, the main issue is the large range of outcomes in patients. So some studies have shown a sort of 20-fold difference in outcomes. So some people actually can have no problems with this treatment, and others have a really large loss in lung function and lots of side effects from it. And there's currently no way really to predict which patient is going to be severely affected. So this data set actually is patient data from the data set we're working with. So 100 patients, and on this axis we've got the forced expiratory volume in 1 second, so that main clinical measure I mentioned at the start, showing percent predicted. So 100% is what a healthy measurement should be. Here, this is the plotting the change in lung function before and after the radiation therapy. So you can see in here, in fact, some patients actually have an improvement in lung function, and this is thought to be because the tumour may be blocking or pushing on airways and actually restricting lung function. But most patients, on average, will have a reduction in lung function. But you can see that there's a huge variation, so varying from a loss of 40% up to a gain of 40% in lung function. And at the moment, there's no way to predict which patient is going to have these outcomes. And one of the important aspects is the fact that most of these patients have a history of smoking, so they have comorbidity, so they often have emphysema. So if you look at the CT scan, this sort of tissue here looks kind of normal. I think it's quite hard to see on here, but if you look up here, you can see signs of emphysema, where you can see a largening of the air spaces in there. So this will impact the lung function and the effects of the treatment as well. So the vision with this work is that we're aiming to develop an in silico or computational modelling platform that can help to predict patient outcomes after treatment. So we are working towards this. So at the moment, we're working really on this step, so predicting the baseline function. So we're creating patient-based models, simulating their ventilation at the moment, but eventually perfusion, gas exchange, and those sorts of functional measurements. And then we'll be able to apply the radiation treatment plan to the model. So therefore, using the simulation to predict or apply the radiation therapy and predicting the post-lung function outcome. So we hope that with this, the clinical outcomes or the application for a clinician is that they will have help in deciding whether they should treat this patient. So how much lung function will they lose? Is it going to be too much? Should they not be treated? Can the dose be escalated? So it's been proven that higher doses are more curative, obviously only up to a certain point, but lower doses are used when you want to be a bit more cautious. And can the dose distribution be improved? So there's techniques being tested where they can preferentially irradiate already damaged tissue and actually improve outcomes for patients. So as I mentioned, we're working with UCL. We have a data set of about 100 patients before and after radiation therapy. So this consists of CT scans and lung function measurements. So in the modelling pipeline, so far we've created 25 patient-based models. So we're using the models developed at ABI in the lung group, initially to fit finite element models of the lung surface. Then within here, we generate models of the conducting airways. So we can extract information for the large airways from the CT scans. We then use a volume filling branching algorithm to generate smaller airways that you can't see in CT scans. So these are realistic airway tree models and are patient-based in terms of some of the parameters from CT. In terms of function, at the moment, we're just simulating ventilation. So this model was developed by Annalisa Swan, and it's sort of referred to as the balloon on sticks model. So we use the mathematical equations that represent fluid flow through the airway tubes. But we also have these sort of balloon-like structures on the end, which represent the gas exchange tissue. So these basically expand based on the mechanical properties of the lung tissue and change the pressure in that system, and we then solve for the flow through these airways. So there's sort of a balance between the resistance in the airway tubes and the mechanical properties of the acinar tissue. So to simulate function in these lung cancer patients, we're altering the model in 3 different ways. So we can segment the tumour location in a patient-specific way. So we then add that tumour into the model. We change the compliance of the acinar tissue in that region, so we make it very stiff. So there's a tumour there, it's not going to expand much during breathing. We also constrict or partially constrict airways that are within that tumour volume, so mimicking the fact that the tumour will be pushing on the airways. And we also include emphysema in these patients. So we can extract from the CT scans, based on the density values in the CT scans, we can see where the emphysema is in a patient-based way. We're then including that into the model as well by changing the compliance of the acinar tissue. So we can also then, we have the dose map for each of the patients. We can apply that personalised dose map onto the computer model. So the study that we've done so far had the hypothesis that we could use the ventilation within that dose volume as a predictor of patient outcomes. So for example, for each patient, we simulate the ventilation in their personalised model. We then sum the airflow within this dose volume, and then look at, if we were to remove, for example, remove all of that tissue, how much function will we lose? This graph here is showing the amount of ventilation. So for the 25 patients, this is the amount of ventilation within the dose volume. So as we go across here, we're getting larger airflow in the dose volume, and this is the measurement, their clinical measurements of the change in lung function after treatment. So, I mean, it seems fairly obvious, but the patients that have higher ventilation in their dose volume are actually having a larger decline in lung function. So this is still work that's ongoing, so we aim to make personalised models for all 100 subjects, but there's other factors that we need to bring in here, such as the damage that the radiation therapy is causing to the lung. So the second aspect is related to COPD, this chronic obstructive pulmonary disease. And here are the summer student up there, had a poster on this actually, so it's still up there if you want to find out some more. So this work was part of Flagship 6 in the MedTech core with Robin Whitaker and Rosie Dobson. So they're developing a mobile pulmonary rehabilitation app. So pulmonary rehabilitation is a sort of formalized program that includes exercises, education, and breathing techniques. So things like relaxation breathing, but also breathing to try and clear mucus out of patients' lungs. So this is for patients with chronic respiratory disease, and it has been shown to be very effective. So with COPD in particular, there's actually not that much treatment that can be done for the patients, but PR has been shown to be very useful. But the problem is there's very low adherence. So a lot of people can't make it to the clinics or don't make it to the clinics for various reasons. So we're developing, they're developing this mobile pulmonary rehabilitation app so that people can do it from home. And so far they've tested this on 26 patients and they've had very good feedback. So the part that we're involved in is more of the educational aspect. So we've been using our lung models for education. So we've made a movie, so this is on YouTube. We've used the models to really describe how the lungs work and some of the changes that are occurring in diseases. And this here, you've probably all seen this shown somewhere. This is our model-based lung app. So the patients were sent a text message which had a link to this app so that they could go and look at it and have a play around to really understand how the lungs work. The other aspect of COPD is related to MR imaging. So we've been developing functional MR methods at the Centre for Advanced MRI. So ultimately we want to be able to measure perfusion and ventilation. So far we only have the ventilation imaging working. So with this technique, it's normally very difficult to see inside the lungs. So you typically get quite a dark image because there's so much air in there. So we're using oxygen as a contrast agent. So oxygen is paramagnetic, meaning that when oxygen is breathed in, you get a brighter signal in the MRI machine. So in fact, the participants, so they wear a face mask in the scanner and they breathe, they switch between breathing air and oxygen. And basically what we're measuring is how quickly that oxygen goes in and out of the lungs. So units that have or get brighter faster, or the oxygen is washed in faster, have a higher ventilation. So we get these ventilation maps, so far only in a single slice. So we have tested this in 8 healthy volunteers, in fact a few from ABI, over 60 years old, and we've compared that with 4 patients with idiopathic pulmonary fibrosis and 4 patients with COPD. And this is still work ongoing, but I just wanted to show this one graph. So what's quite interesting so far, so this is the measured FEV1. So we had the volunteers and the patients have their lung function measured. And on here, this is the coefficient of variation of the ventilation measurement. So a measure of the heterogeneity of ventilation. And what's quite interesting, so the red ones are the healthy volunteers. They have a heterogeneity of about 50%. These patients with fibrosis also have a heterogeneity that looks fairly healthy. So actually when you look at their ventilation maps, it looks fairly healthy, even though they do have a lower lung function. Whereas you can see the COPD patients up here have almost twice as much heterogeneity in their lungs due to the disease effects. So finally getting on, save the best for last, getting on to e-cigarettes and vaping. So this is quite a new project. We started it last year. So you've probably all heard of these now. They've been in the news a lot the last few years. So they're originally developed as a smoking cessation tool. So with the aim that they will be a healthy alternative to cigarettes. And there have been some studies showing that they are an effective smoking cessation aid, but this is not conclusive yet. Some studies have shown that they're no better than normal smoking cessation aids, so patches or Nicorette gum. So that's still being looked into, but the other negative effect is that a lot of cigarette users are now using both products. So they're using e-cigarettes and cigarettes, so they're having potentially twice as much damage occurring in their lungs, but also there's a lot of younger people or non-smokers starting to use these products, and particularly in the US. So these are just some headlines that I copied. So you would have probably heard the news from last year. So there was this big outbreak of lung disease. So they've termed this EVALI, which is e-cigarette or vaping-associated lung injury. And so far, there's been about 2,700 hospitalizations and 60 deaths, but this number is still increasing. And this is actually thought to be most of the people admitted to using THC or cannabis e-liquids, and they're thought to be sort of more of the bootleg products where they have added vitamin E acetate, so they've added that as a thickener to the product, and it's sort of poisoned people's lungs. But it sort of just shows the danger of this. So at the moment, there's no regulations on e-cigarettes. They don't have to list what's in the liquids, and you've probably noticed that they're allowed to be advertised on TV or on billboards. So an e-cigarette works by using these sorts of devices. So this is a tank-type e-cigarette. You put the e-liquid in here. There's a metal coil that sits in there, which is battery-operated, and it heats the e-liquids to very high temperatures until it vaporizes or forms an aerosol that the user then inhales. This is more of a pod-type e-cigarette. This is actually a Juul, so those are the ones that have become very popular in the US, especially with the kids. And I watched a documentary on it, and it's because they're very discreet, so they just look like pen drives. They don't produce a very big cloud of aerosol, so kids can actually put them in their sleeve and use them when they're in class. The other aspect of these is that Juul developed these nicotine salts, which meant that they could make very high concentration nicotine e-liquids. So each of these pods, which is only about a centimetre cube almost, contains equivalent to a packet of cigarettes. So a normal cigarette is about 12 milligrams of nicotine. They can make these up to about 60 milligrams of nicotine. So they're getting, the kids who are starting to use them are finding that they're very addicted to nicotine and they can't give it up. So the e-liquids contain propylene glycol, glycerine, nicotine, and flavorings. But as I said, there's no regulations that require a listing of the ingredients on the packet. So basically anything can be added for flavoring, for example, or anything else. So lots of dangerous chemicals have been found in the aerosols of e-cigarettes. So even some of the flavorings that are added often are thought to be safe for ingestion and not safe for inhaling. So even things like vanilla flavoring or cinnamon flavoring or buttered popcorn has been found to have very serious effects on the lungs. Other things that have been found, formaldehyde has been found in some studies, other carcinogens, but also heavy metals. So the coils or any of the metal material within the device, because of the high temperatures, that also vaporizes and has been found to go into the aerosol. So the FDA have made these cool posters. I really like this one. You may not be into chemistry, but if you vape, chemistry may be very into you. So the warnings are out there, but nobody's going to know the long-term health effects for another 10 or 20 years, so it's going to be very difficult. But I feel like it's history repeating itself from cigarettes. When they came about, people had no idea they were negative. They became too widespread before they could be controlled, and I feel that the same is happening here. So our plan is these four main aspects. So we're wanting to see what is in the aerosol. We're doing chemical analysis of the aerosol with different flavors, different concentration of nicotines, different devices. Where does this aerosol go? We plan to do particle transport modeling, so simulate where the particles go and where they deposit in the lung. And we're going to link that to some experimental work where we will expose epithelial cells and lung tissue to the aerosol and look at the response of that. And we also plan to do some MRI studies. So far we have had 2 people where we've taken an MRI of the lung function before and immediately after they vape. So this is an example of the rig that we've developed already. So we can add any e-cigarette here. We can create the aerosol. Within here we've got some cells in here that we can expose the aerosol to them, and we can collect the condensate. And the chemical analysis that we've done so far is using gas chromatography, mass spectrometry. So this is at the Chemical Engineering Department. We've found over 50 different chemicals, most of them flavorings. but this is work that we want to continue. But we've also found these heavy metals in here. So we've found high levels of lead, zinc, copper, and nickel. And I'm finished. I think I've done it on time. So thanks, everyone.